Hello students, welcome to the EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor at Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. So today we are going to talk on lipases, the most promiscuous enzymes from the paper Structure and Function of Biomolecules 2. We recognize that when it comes to specificity, not all enzymes are created equal. We are normally introduced to this concept of enzyme specificity with the illustrated example of proteases, and we learn there that the trypsin is highly specific, chymotrypsin somewhat less so. Proteases like papain and ficin are fairly non-specific. Enzymes also show surprising amount of non-specific binding. Textile dyes are known to bind to variety of enzymes. Aptamers, peptides, and many polymers bind to the enzymes. Non-specifically, but fairly firm binding, and that imparts their binding a reasonable amount of selectivity, and these facts are exploited in protein purification. So it's not as if the molecular recognition processes involving enzymes are absolutely very specific. So in this module we will discuss some consequences of the deviations from the specificity in an extreme way, and this behavior is termed as enzyme promiscuity. So the objective of this module are to understand what is enzyme promiscuity, what do we mean by it, and then to learn about different classes of enzyme promiscuity. And since basically we are talking of all this in the context of the lipases, we have to see that first we learn about the broad specificity of the lipases, and after that we learn about the promiscuous reactions of lipases. So the concept map is very simple, that we learned about enzyme promiscuity in general, and substrate promiscuity, catalytic promiscuity, and condition promiscuity as its three classes, but we discuss all this in the context of the promiscuity of lipases, because this whole paper is about lipids. So we will show that lipases can act upon substrates which are neither fats or oils or esters, that they can catalyze unexpected reactions such as aldol condensation. This phenomenon of enzyme promiscuity is obviously of interest to organic chemists, as it enables use of lipases in synthesis of even more wider range of products. Lipases are not the only class of enzyme to show this phenomenon of promiscuity, hence we start with a general discussion and then move on to illustrations which focus on promiscuity of lipases. While introducing enzymes as biocatalysts, it is normal to talk of their specificity. Specificity in fact could be of various kinds: substrate specificity, regiospecificity, and stereospecificity. As far as substrate specificity is concerned, it is also normal to add that some enzymes are very specific. We have given frequently the well-known example that trypsin will selectively cleave peptide bonds involving arginine or lysine residues. On the other hand, there are other proteases itself like papain or facin which have somewhat more broad specificity. Actually, our idea of substrate specificity is introduced via enzyme commission classification of enzymes. So, all the three examples cited above are hydrolases and will have the same first number. There will be a common X after EC. What is more, since they all belong to the subclass of proteases, they will also share the second letter Y. Whatever is the number assigned to the proteases will be the Y in this case. So the EC classification is based upon reaction type and not the substrate specificity of the individual enzymes. Appreciating this will help us in understanding this phenomenon of enzyme promiscuity. During last several years, a large number of CC bond formation reactions catalyzed by lipases have been reported. Such reactions are very useful in organic synthesis. So this promiscuous behavior has caught the attention of biochemists. Lipases classified as hydrolases in such reactions actually show a synthetic activity. In fact, such behavior had been noticed by biochemists for a long time in sporadic cases. Both lipases and proteases have been known to show also another kind of a hydrolase activity called esterase activity. Thus, lipases are assayed by both triglyceride hydrolysis, the well-known tributyrene hydrolysis assay, normally tracked by changes in pH using a pH stat. We also assay lipases by ester hydrolysis, which is a spectrophotometric assay which tracks the hydrolysis of a simple synthetic ester like para-nitrophenyl butyrate. This was clearly understood in terms of their reaction mechanism and active site geometry. It was also rationalized by pointing out that this can be treated as a case of broad specificity, and of course, since both were hydrolase activities. Perhaps well-known oxygenase activity of Ribisco was not considered a reason to rethink our notion of enzyme specificity. It is interesting to note that oxygenase activity of Ribisco has been considered as an outcome of evolutionary history of the enzyme. It is believed that when Ribisco evolved, there was no oxygen in the atmosphere. It seems that later on evolution did not correct for this accidental promiscuity which leads to photorespiration. It is worth noting that carboxylase and oxygenase activities are two different reaction types and have different EC number. Principle of microscopic reversibility states that reactions can be reversed at the molecular level. Consider the following ester hydrolysis by a lipase, which is shown up there in this slide. So we have an ester called RCOOR dash being hydrolyzed to form RCOOH acid and alcohol, which is R dash OH. So for this reaction we would start with an ester. We normally use excess of water to make sure that adequate hydrolysis occurs. But suppose we start with the products on the right hand side instead and carry out the reaction in the absence of water. In module 29, we will learn several ways of doing it, say in excess of R-OH itself. In such cases, it has been shown that lipase catalyzes the reverse reaction and the ester is indeed formed. As this requires a different reaction medium, it has been suggested that this is simply called condition promiscuity and this be included as a class of enzyme promiscuity. Condition promiscuity is shown by many enzymes, of which lipase is actually an important example. Enzymes may use the same chemistry on a different substrate during their catalysis, of course. This kind of a behavior is termed as substrate promiscuity or substrate ambiguity. One estimate shows that, as one would expect, substrate promiscuity is more widespread than catalytic promiscuity, which is also sometimes called reaction promiscuity. This has been in classical enzymology referred to as broad specificity or relaxed specificity. One can also include coenzyme promiscuity under substrate promiscuity. Coenzyme promiscuity can be exemplified by a dehydrogenase being able to work with both NAD or NADP coenzymes to a varying degree. So lipases show very high promiscuity of the substrate kind as they accept wide range of esters during their esterase activity. Enzymes in their wild form, that is without any mutation done by using rDNA techniques, they show all the three kinds of a promiscuity. The three kinds of a promiscuity shown by many lipases are further illustrated now. Protein engineering has been used to generate or improve existing enzyme promiscuity. So, serine 105 ELA mutant of CalB showed 100 times higher specific rate for some CC bound reactions. This is generally referred to as induced promiscuity. At one time, it was believed that catalytic rates of promiscuous reactions are slower and those observed during normal catalysis than those observed during normal catalysis. So, more recent data shows that the rate acceleration up to 10 raised to the power 18 have been observed even for promiscuous reactions. Conformational flexibility is believed to be an important parameter which decides the probability of the enzyme being involved in catalyzing a promiscuous reaction. So, the conformational flexibility around the active site is especially believed to play an important role. Hydrophobic bonds are dependent upon desolvation and are entropically driven. These are not so much dependent upon stereochemical fit between these two entities. Hence, substrate hydrophobicity facilitates catalytic promiscuity. Optimum pH is a well-known characteristic of the enzymes. In some cases, the optimum pH of an enzyme in a normal reaction and promiscuous reaction can be different. This is because the two reactions may be facilitated by different protonation states of the same catalytic residues. Substituting different metal ions is known to induce promiscuous activities. Of course, since lipases are not metalloenzymes, this is not of direct relevance to lipases. It is believed that the primordial enzymes were highly promiscuous, as fewer enzymes were expected to help the organism to survive. Divergent evolution via gene duplication and mutations were driven by selective pressure to evolve more specific enzymes. Before we discuss promiscuous reactions of lipases, let us first familiarize ourselves with specificity of lipases. Structurally, all lipases are part of alpha beta hydrolase fold family. In these structures, the core of parallel beta strands is surrounded by alpha helices. The active site of lipase consists of a catalytic triad of serine, histidine, and third residue is an acidic amino acid side chain, which can be either aspartate or glutamate. Just like serine proteases, here also serine is the key active site residue here. The triad which we mentioned is also reminiscent of the way the proteases function. So it is not surprising that both proteases and lipases show actually esterase action, albeit the kind of esters which they hydrolyze or form are of different structural nature. But coming back to the active site serine, this nucleophilic serine is at a hairpin turn between a beta strand and alpha helix and is a part of highly conserved pentapeptide sequence gly X, serine X and gly. The X in this sequence of course means that while the other three residues are fixed, X can have somewhat, X position can have some little latitude and those amino acids can be different. The only exception is CalB, which does not have this pentapeptide around the active site. CalB is a very frequently used lipase, particularly as we will talk about it elsewhere when we give some illustrative examples of the use of the lipases in synthesis or otherwise, we will frequently come across this enzyme. So this particular lipase is a very extensively studied and used enzyme. As fats and oils are the natural substrates of lipases, that is the role which lipases play during their digestion, that they hydrolyze fats and oils. Those fats and oils we consume as diet. So it is common to talk of regiospecificity of lipases, acyl chain on the 3 carbons of glycerol. Many non-specific lipases hydrolyze ester bonds at all the 3 carbon atoms of the glycerol. So such lipases are called non-specific lipases, and the term non-specific is restrictive in the meaning that these lipases do not distinguish between the ester bonds which are present at either the terminal carbons of the glycerol or the middle carbon atom. So in such cases, diglycerides and monoglycerides are intermediates, but these do not accumulate. The reason is that these are hydrolyzed quite often, even more rapidly than triglycerides. So in a way, one could say that the lipases also have diglycerides and monoglycerides as substrates, and this actually is very important in biotechnology because both diglycerides and monoglycerides are extremely useful biosurfactants in many industries particularly the food industry. On the other hand, there are other lipases which are 1,3 specific. That means these lipases release fatty acids from the fatty acid ester bonds at terminal positions of the glycerol 1 and 3 positions. These also give monoglycerides and diglycerides, and since the terminal positions are identical and the ester bond at the two position is not attacked, the 2 monoglyceride accumulates in such cases. In the lipase nomenclature, sometimes the name of the microbe, the source microbe has changed, and that sometimes can get confusing. For example, Pseudomonas cepacia has been described in the early literature extensively, and sometime people continues to still call it Pseudomonas cepacia lipase. However, the current nomenclature, instead of calling the microbe Pseudomonas cepacia, calls it Burkholderia cepacia. So it's better to call this lipase as a lipase from Burkholderia cepacia lipase. Burkholderia cepacia lipase is a non-specific lipase. So is the lipase from Candida rugosa. These are widely used lipases, that is because they are commercially available from quite a few vendors. Another lipase which is also a very important non-specific lipase is the lipase from Thermomyces lanuginosus. The catalytic promiscuity of lipases consists of formation of C-C bond, C heteroatom bonds, heteroatom bonds, and even oxidative processes. C-C bond formations by aldolases are very useful synthetic reactions in organic chemistry. Aldol condensation has been reported to be catalyzed by several groups. Both Calvi and porcine pancreatic lipase has been found to give excellent conversions, albeit with long reaction times. Limited solubility of many aldehydes in aqueous buffers results in most of the reactions being carried out either with very small concentration of aldehydes or adding a small percentage of water soluble organic solvents. If one of the substrate is a water miscible organic solvent, this problem does not arise. However, a large amount of water miscible organic solvents results in lipases being inactivated. Hence, optimization by considering various trade-offs is very necessary. The medium also plays an important role in controlling enantioselectivity selectivity even for these catalytically promiscuous reactions. The reaction between para-nitrobenzaldehyde and acetone catalyzed by lipases illustrates this. With 20% water, 96% conversion and 15% EE was observed. In nearly anhydrous medium, percentage EE improved to a value of 44%, but significant drop in percentage conversions was reported. This results as well as quantum molecular modeling in the context of aldol reaction catalyzed by Calvi indicate that the active site serine is not involved in the catalysis of these C-C bond formations. Presumably, histidine acts as the base. It is not clear why replacing serine accelerates the catalytic rates. The lipase from mucomehi has been shown to catalyze one-pot Manish reaction between acetone, aniline, and various aromatic aldehydes. The reaction medium was an aqueous buffer. Michael addition between secondary amines such as pyrrolidine, piperidine, diethylamine, and acrylonitrile illustrates the formation of other types of bonds catalyzed by lipases. They catalyze preparations of peroxycarboxylic acids. The direct epoxidation of alpha beta unsaturated aldehydes, butenol and 3-phenyl-proptoenol by using H2O2 has also been possible. Synthesis of heterocycles is another useful type of reactions which has been catalyzed by lipases. The reaction between fatty acid esters and 1,2-aryline diamines led to the synthesis of 2-alkyl benzimetazoles. Similarly, synthesis of dihydrofuran by using lipase as a catalyst has been possible. Again, such instances are not limited to use of one specific lipase. For example, both mucomehi lipase and porcine pancreatic lipases have been used in synthesis of heterocycles. Slide 17. Three-component Hench reaction has been catalyzed by Calvi to obtain 1,4-dihydropyridines with 4-nitrobenzaldehyde, acetylacetone and acetamide as the reactants. 90% yield was possible in 72 hours. Lipases do not prefer omega fatty acids as substrates. Omega-3 PUFA, eicosapentaenoic acid and DHA are important fatty acids. Their presence as fatty acids in oil is considered desirable from nutritional perspective. DHA occurs in fish oils. The DHA concentrates have been obtained by hydrolyzing other oils by lipases in aqueous medium. Extensive work on enrichment of omega-3 PUFAs in fish oil by lipases has been carried out. The synthetic utility of lipases has been exploited at the industrial scale as well. BASF process for obtaining chiral amines involved selective acylation of one enantiomer present in the racemic mixture of amines by using alkyl methoxy acetate as the acyl donor. Another interesting type of application of lipases is to prepare esters from alcohols and acids, each of which has some useful property. Ruthene and vanillyl alcohol are antioxidants. These were esterified with omega-3 fatty acids from fish. Narangine was converted to narangine 6-racinoleate using castor oil itself as acyl donor. L-ascorbic acid was used to esterify cinnamic acid to create a more powerful antioxidant. In many of these applications, lipases have been used in immobilized form. Immobilized enzymes have a long history of applications in industrial enzymology. Most of these applications have been in aqueous media. In case of low water media, even free enzymes are insoluble, hence reusability is not an issue. The major advantage of immobilization such as media is that mass transfer is improved. Free enzyme will be in the form of a precipitate or powder and will act as a heterogeneous catalyst. After immobilization, enzyme is spread over the surface of the support and offers larger catalytic surface. This results as well as quantum molecular modeling in the context of aldol reaction catalyzed by Calvi indicate that the active site serine is not involved in the catalysis of these CC bond formations. Presumably, histidine acts as the base. It is not clear why replacing serine accelerates the catalytic rates. The lipase from mucomehi has been shown to catalyze one-pot Manish reaction between acetone, aniline and various aromatic aldehydes. The reaction medium was an aqueous buffer. Michael addition between secondary amines such as pyrrolidine, piperidine, diethylamine and acrylonitrile illustrates the formation of other types of bonds catalyzed by lipases. They catalyze preparations of peroxycarboxylic acids. The direct epoxidation of alpha beta unsaturated aldehydes, butenol and 3-phenyl-proptoenol by using H2O2 has also been possible. Synthesis of heterocycles is another useful type of reactions which has been catalyzed by lipases. The reaction between fatty acid esters and 1,2-aryline diamines led to the synthesis of 2 alkyl benzimetazoles. Similarly, synthesis of dihydrofuran by using lipase as a catalyst has been possible. Again, such instances are not limited to use of one specific lipase. For example, both mucomehi lipase and porcine pancreatic lipases have been used in synthesis of heterocycles. 3-component Hensch reaction has been catalyzed by Calbi to obtain 1,4-dihydropyridines with 4-nitrobenzaldehyde, acetylacetone and acetamide as the reactants. 90% yield was possible in 72 hours. Lipases do not prefer omega fatty acids as substrates. Omega-3 PUFA, eicosapentaenoic acid and DHA are important fatty acids. Their presence as fatty acids in oil is considered desirable from nutritional perspective. DHA occurs in fish oils. The DHA concentrates have been obtained by hydrolyzing other oils by lipases and aqueous medium. Extensive work on enrichment of omega-3 PUFAs in fish oil by lipases has been carried out. The synthetic utility of lipases has been exploited at the industrial scale as well. BASF process for obtaining chiral amines involved selective acylation of one enantiomer present in the racemic mixture of amines by using alkyl methoxy acetate as the acyl donor. The ring opening of an oxazolene with butanol leads to the synthesis of L-tertiary leucine. The reaction medium was toluene and lipase from mucomehi was used as the catalyst. Lipase can substitute for phospholipase A2 and A1 in conversion of phospholipids. Such conversions have been carried out in toluene, hexane or even solvent-free media. The starting material is a naturally occurring lipid to obtain a specific glycerophospholipid. Many 1,3 specific lipases show specificity towards and SN position of glycerophospholipid, a type of reaction which would normally require phospholipase A1. As polysaccharides like starch are renewable materials, there has been a considerable interest in industry in acylation of starch. An interesting approach has been to use Novozyme 435, which is a lipase, as the biocatalyst to acylate starch nanoparticles with vinyl acetate. Novozyme 435 is in fact the immobilized form of Calbi. Another interesting type of application of lipases is to prepare esters from alcohols and acids, each of which has some useful property. Rutein and vanillyl alcohol are antioxidants. These were esterified with omega-3 fatty acids from fish. Narangine was converted to narangine 6-racinoleate using castor oil itself as acyl donor. L-ascorbic acid was used to esterify cinnamic acid to create a more powerful antioxidant. In many of these applications, lipases have been used in immobilized form. Immobilized enzymes have a long history of applications in industrial enzymology. Most of these applications have been in aqueous media. In case of low water media, even free enzymes are insoluble, hence reusability is not an issue. The major advantage of immobilization such as media is that mass transfer is improved. Free enzyme will be in the form of a precipitate or powder and will act as a heterogeneous catalyst. After immobilization, enzyme is spread over the surface of the support and offers larger catalytic surface. So, let us summarize what we have learnt in this module today. We learnt about a phenomenon called enzyme promiscuity, which basically tells us that the enzyme sometime can catalyze reactions which is not obvious from their class of to which they belong. We learnt about the various kinds of enzyme promiscuity, and we learned few examples of promiscuous reactions catalyzed by lipases. So in the case of many lipases, immobilization also offers another advantage. Many hydrophobic and in some cases even hydrophilic surfaces, when used as support, results in opening up of the active site molecular lid, which is normally a peptide. This also happens when lipase encounters a surface. Hence the phenomenon is known as interfacial activation of lipases. This enhances lipase activity several times. Lipases top the growing list of enzymes which are now used in organic synthesis even at the industrial scale. Both their broad specificity and catalytic promiscuity continue to power their synthetic applications. Thank you.